Okay, so I did my project on a research project on brain-derived exosomal proteins as effective biomarkers for Alzheimer's disease. Um, Here's a brief table of contents that I'll be going over. What is Alzheimer's disease, AD, about the project, the testing process, the results, and the future implications. So one, what is Alzheimer's disease? Senile dementia, also known as Alzheimer's disease, AD, is a progressive neurological disorder that slowly destroys and kills the brain connections and cells that affect a person's memory and other crucial mental functions. The disease affects over 50 million people worldwide and becomes more common at ages 65 and older. The cause of AD is currently unknown and there is no known cure for the disease. However, early diagnosis can lead to more opportunities for effective treatments. About this project. Who made this happen? There were three scientists/researchers that were involved in this project: Kai-Young Kim, Ki-Young Shin, and Keon A. Chang. And some other information includes the location, which was in Korea, the funding by the Korean government, and the National Research Foundation Bio and Medical Development Program. The current project status is complete. So down to the actual project itself. So current factors to AD diagnosis is blood, saliva, cerebrospinal fluid, also known as CSF, structural MRI, and a PET. The key factors to the diagnosis is CSF, which is rarely performed by general practitioners due to the nature of the procedure. This is where the usage of blood analysis comes in. Blood analysis, a lab analysis found in blood in AD, has shown to be extremely promising in all aspects of AD testing. Additionally, blood collection is minimally invasive, which is where we begin to discuss the use of blood biomarkers, a biological molecule that is found in blood and is a sign of regular and irregular process. Since blood biomarkers would prove to be a huge aid to early AD diagnosis. With all of this in mind, utilizing BD proteins, extracellular nanovesicles released by all cell lineages of the central nervous system that are detected in the peripheral blood, this project proposes to evaluate those BD proteins in the peripheral blood as blood biomarkers for AD by administering standardized analysis with meta-analysis to discuss the future usage of BD proteins as AD biomarkers. So some benefits to this project, beneficiaries to this project are victims of AD and their respective families and neurology researchers. If victims of AD can discover the disease early on, it will lessen anxiety and victims can participate in clinical trials for treatments for Alzheimer's. The families of many victims to AD can also prepare for the financial and communal effects of AD, with more early on cases for researchers to study conclusions to what causes the disease and more information on early AD and what can be done to potentially prevent AD can be discovered within these clinical trials. So the testing process, results, and the future implications. So the researchers first chose 20 articles of 342 to be used as eligible studies and previous works to look back on when studying their work. These studies showcased the BD proteins for AD. Then researchers tested these BD proteins from the articles for AD. And continuing this, thirdly, researchers tested the changes of exosomal proteins in AD. Continuing, researchers tested the proteins in AD and whether their levels increased, decreased, or remained the same. From this, researchers then chose 6 proteins to use in the meta-analysis. Here are all 10, or it might be more proteins, but about 10 from what I counted. The last test was using meta-analysis of duplicated BD proteins in AD, and 6 proteins were used for this final test. And here are the results from all 6, and I'll explain furthermore in the next slide. So the results from all these tests from the 6 final proteins that were chosen from all previous tests have inconsistent results, unfortunately, on whether they can be effective in AD diagnosis. However, 4 of the 6 have been suggested as potential effective biomarkers and would need more research and time to fully be boot from the entire discussion. And the remaining 2 have also been suggested for consideration in future research, even though the results from those 2 weren't significant. So future implications, while the results were inconclusive, it doesn't fully close the case on whether BD proteins are effective, and results will, for this will need more time and testing to get a clear-cut answer on how they can be of use. Here are my citations, and thank you. And if you have any questions, you can email me down below.